Well, welcome everyone to this new session of the SFPM seminars. Today we have the honor to have with us Zinaida Perova. She completed her doctoral studies at the Coldstream Harbor Laboratory, followed by postdoctoral positions at the MRC Laboratory of Molecular Biology and the UCL. In 2018, she joined the EMBL's European Bioinformatics Institute as a data scientist to work on the Human Cell Atlas data coordination platform. Since 2020, she has led a team of developers at the EMBL EBI to deliver cancermodels.org platform. And currently, this is the largest open catalog of harmonized patient-derived cancer models. She is actively involved in several international collaborations and co-leads the GA4GH cancer community. So, Zina, thank you so much for being here today, and the floor is yours.
Thank you so much, John. And Aaron. So I am going to share my slides. And I am going to close the Zoom windows, so I won't see anyone's hands, but please feel free to interrupt if you have an urgent question throughout the presentation. So I'm very, very grateful for this opportunity to present our platform, cancermodels.org, and tell you a little bit about how we see the precision oncology can be enhanced through the collaborative cancer models resources. So just a quick overview. I'm coming from the research background, having worked on animal models and then transitioning to the services and to now creating solutions for the researchers and clinicians working with these models. So it's kind of, this is one quick background and where I'm coming from personally. And I've worked on several projects, as mentioned, Human Cell Atlas, PDX Finder, now cancermodels.org. So patient-derived cancer models probably do not need a very long introduction to this audience, but in an interest of what I'm going to be describing about our platform, I'll just say that the main groups that they're categorized into that I use and we use in our platform is the 2D models, the patient-derived cells, the 3D models, patient-derived organoids, and also patient-derived xenografts when the tumor of the patient is implanted into the immunodeficient mouse and allows to grow, as opposed to the in vitro models done in either 2D or 3D environment. So all these models have their own advantages, and they bring tremendous value as they allow to study tumor biology and evolution, mechanisms of drug response and resistance, preclinical drug development. They're a powerful platform for functional precision oncology in academic, pharma, and clinical contexts. And I would also want to mention kind of the traditional precision oncology approach of taking a human sample to perform genomic sequencing, to identify actionable targets and treat patients with matched drugs, that over years through large genomic studies showed that most of genomic drivers actually are not clinically actionable, and that only a subset of tumors harbor genomic features that can be used to guide therapy selection. And then genomics doesn't reflect all aspects of the tumor biology. So here comes the functional precision oncology approaches that do not only analyze patient tumors for static features, such as the presence of targetable mutations at a given time, but also utilize the patient-derived models that allow evaluation of drug responses and functionally testing them and then treating patients with matched drugs. So functional precision oncology allows drug sensitivity testing within patient-specific genetic and epigenetic background, prediction of drug sensitivity for therapeutics without genomic biomarkers, and also tremendously expands treatment options for those patients who exhausted their standard of care treatment. The use of patient-derived cancer models and functional precision medicine approaches has been continue to grow, as indicated by the number of publications, current as well as projected, and also by the recent incorporation of these approaches into clinical trials. You can see that the two trials here that were done in Europe in 2022, and one example of the recent trial in the US, and all these showed an improvement in either with previous therapy or in progression-free survival for those patients who have been matched with drugs using the functional precision oncology approaches. So these models are generated now by contract research organizations, by academic labs, by pharma, institutional biobanks, providing researchers valuable tools to test their hypotheses, however, not without challenges. So, and let's look at one use case. This is Sam. Sam is a clinical researcher working on colorectal cancer. Sam would like to find patient-derived models with KRAS and TP53 mutations. So she goes online to search for models that exist, and she found 588 models, in vitro models, in the Human Cancer Models Initiative's searchable catalog, 422 models in another resource, the NCI's Patient-Derived Models Repository, and over 2,000 cell model passports. So here comes challenge number one, which is multiple platforms that provide information about these models that exist. The challenge number two is heterogeneity of the attributes that these platforms show. So you can see that for a human, some of them are, you can figure out what is what, how similar they are. But some of the attributes you can't even filter on in certain platforms, and it takes 2 clicks away to actually find these details, while in the other platform, all this information is available and you can actually filter on specific parameters. The next challenge is the heterogeneity of metadata. And as you can see here from the values that these attributes have, there is heterogeneity in the level of granularity, as indicated by this example, definitions, as well as semantics. In one platform, in the example of diagnosis, diagnosis is mapped to NCI thesaurus. In another platform, it would be mapped to the subset of a MEDRA ontology. And in the third platform, there is an internal vocabulary that is used to depict the diagnosis. So again, for the human, if you're comparing 3 models, you could probably deal with this, but on a larger scale, this heterogeneity becomes a big hurdle. And the fourth challenge is, in addition to that, there is also different data access mechanism. In one platform, Sam can get all the data as a collection of TSV files. In another platform is one file containing all information about the models of interest in CSV format. And in a third platform, it requires programmatic access to actually obtain metadata of the models of interest. So you can imagine that it's going to take quite a while for Sam to standardize, harmonize all this information collected from these platforms before actually being able to choose the right model to actually start doing experiments. And this is what we are trying to solve by providing a unified platform that allows to search over patient-derived cancer models from multiple resources in one place, all harmonized, and essentially do this work do this work for Sam. And I would like to show you a quick overview of our platform. This is the video that we made last year, just 1 minute, that will give you a sense of how the platform looks like and works.
Finding the right patient-derived cancer models for research can be daunting. The data is fragmented and scattered, making it hard to find what you need. At cancermodels.org, we solve this problem by standardizing, integrating, and publishing complex data on patient-derived cancer models. Our platform lets you search and compare over 8,000 models, including data on diagnosis, gene mutation, and drug treatment. Our intuitive web interface and REST API allow you to filter and find models by cancer type or molecular data. Plus, our knowledge base links to external resources, offering a comprehensive view of each model. As an open source, community-driven resource, cancermodels.org breaks down information barriers and fosters collaboration. This accelerates cancer research and helps develop personalized treatments, supporting global health equity. Join us at cancermodels.org and revolutionize your cancer research today.
So the mission of cancermodels.org is to accelerate cancer research and improve patient outcomes by providing access to next-generation cancer models that accurately represent diversity and complexity of human cancers. And the way we do it is by aggregating, harmonizing, and standardizing global patient-derived cancer model data from academic and commercial providers, by providing comprehensive information on models, by linking to additional data and resources, and enabling easy navigation to the originating resource for models and data. The impact is easily accessible, comprehensive information on patient-derived cancer models and associated data, and subsequently, advanced understanding of tumor biology, facilitated anti-cancer drug development, and personalized treatments. So I'd like to give you the quick project overview and how specifically we address the challenges highlighted in the first part of my talk. So we provide harmonized attributes and standardized metadata from multiple resources that have cancer models. We map to ontologies to enable seamless searching across all the resources. We provide a single platform with over now 10,000 models from multiple worldwide providers, and also a REST API for programmatic access to all the data, which is also available to download from the platform. To give a bit more of an overview of the models that we have, as mentioned, we have over 10,000 models right now, and they are within these three mentioned groups: almost 6,000 PDX models, almost 3,000 cell line models, and a bit over 1,500 of organoid models. 10% of these models are from pediatric patients, about 5% include rare cancers, and about 24% are from ethnic minority backgrounds. And on the right-hand side, you can see the cancer systems that the models in our resource cover. Our providers include large consortia, repositories, biobanks, contract research organizations, and individual projects. And you can see a breakdown per model type in this graph. And currently we have 56 models from 56 providers from all over the world. Most of them come from United States. We also have a large fraction of models from Europe, and also from other parts of the world, several providers from Australia, Singapore, and Hong Kong. And we're always trying to expand more to make the models, our portfolio, as diverse as possible. This is the map of our current providers. As mentioned, we work with large consortia such as PDXNet, Pivot, and EuroPDX, also with national networks such as Italian network, institutional biobanks, and also other platforms and projects that generate large-scale collections of models. And the dotted line include the ongoing collaboration where we're working to add these models into the platform, including from contract research organizations. In terms of the data overview, since the release in February 2023, we have been increasing our data in the platform. And you can see the breakdown per data type. About 57% of all models have gene expression data, 56% gene mutation data, followed by immune markers, copy number alterations, image data, treatment data, both patient and model, and also biomarkers. And in total, we have close to 400 million data points. The breakdown of data according to the model type is presented here. As you can see, majority of the models would have mutation expression and copy number alteration data, and we call these kind of the core data types for our platform because of that. And you can see the technology is used to generate this data. Whole exome sequencing, whole genome sequencing, and RNA-seq for the expression data. So the portal looks like this. So this is the search page where you can search by cancer diagnosis. The results will come as cards. You can further filter these cards by 25 facets across 4 modules, as indicated here. And when you receive the results on the search page, we have this model characterization score that reflects the amount of information available about the model. It doesn't have to do anything about the quality, it's just the amount of information available. And if you open the model page, individual model pages, you can see the differences between what's the green score is in comparison to the yellow score. So there are some values that are not provided, which might result in less overall value of this model for particular use case if, for example, someone is looking for a specific age group. We also provide solutions for variable data visibility. And in this case, if a user searches the model by specific mutation or variant, the model will come up in the search results and will indicate that there is data available. However, when going on the model page, the data wouldn't be possible to view the data within the portal, but a request will need to be made to the model provider. And this is done due to some restrictions on the data visibility, which are either because of data agreements or other reasons. Most of the data, though, is available to view through the platform. All the data in the platform is available for download as well as to view through cBioportal instance, and where we always link to raw sequence data for reanalysis if the user wants it to. So the link will bring directly to the study within the raw data archive. Well, in this case, this is in EGA, which is control access data. So obviously the person would have to ask for the to go through data access committee to get the access to the data, but in case of the data being deposited to ENA, then it will link directly to where the sequence data could be downloaded. We always make it clear where the model comes from. So in this example, you can see in the upper right side, this model is provided by Broad Institute. And we indicate if data is available at additional portals, such as its HCMI searchable catalog, which will link you to the information about this model there. Also, we try to link the users with the model providers for any additional information or requesting the model of interest. So it will link you to the person responsible for sharing additional details on information. And if the model is also available for purchase through distributors, we indicate that as well. When the model is present at other portals and is known by different identifiers, we also link all this information, try to make it as transparent as possible. We introduced the related models feature and filter, where multiple models are generated from the same sample, and it's possible to filter on these models within our filters. And this just shows different examples. So that improves accessibility of related models in case someone's looking for a cell line and a PDX that comes from the same patient, or in case of, let's say, one model developing resistance to the certain treatment and essentially generating a new resistant model. So I've mentioned the external resources that we integrate with, such as EGA and Raw Data Archive. We also integrate with cancer annotation resources, such as OpenCravat, Civic Database, ClinGen, OncoMX, Cosmic, that provide information about the genes and variants and their clinical significance. Within the treatment data, we also link to the compounds for additional information on the compounds used in the drug dosing studies. We link to images in the bioimage archive, the histology images that are associated with the models as well. Another part of the project is accessibility of data through the CBio portal for further analysis. And also all the data in the portal is available for analysis through cloud platforms. And we have a couple tutorials available for the users if they're interested in that. So far I've been talking about models that come from major portals. We also have models from individual data submitters, from individual labs. And in order to make the submission of these models to the resource easier, we paired with the Ontario Institute for Cancer Research to implement their Overture components of the Overture software to create metadata dictionary and metadata validator as our data submission tools. So the metadata dictionary explains what kind of information we collect about the models, and metadata validator allows to check the validity of submissions. So the metadata validator allows to upload the Excel spreadsheet with filled metadata about the model and checks it against the metadata dictionary to show whether a submission is valid or not. In case of an invalid submission, such as shown in this slide here, you can see clearly the field name, the type of error, and also the error message. So the users who try to submit the models get immediate feedback, and the submission process requires less iterations, making it more streamlined. In case of a valid submission, this tool also calculates the model characterization score, depicted here on the right, and also provides information on how to improve it, such as which fields need an entry a value to improve the score. It also positions it with the distribution of all the other models within the resource. And these are the model characterization score distributions for the patient-derived xenografts. So the red dot would indicate where the model falls within this the score distribution, and also for patient-derived organoids and patient-derived cell lines. So the purple dot will indicate where the model is within the score distribution. These scores are calculated based on the minimum information standard. So for PDX models, the minimum information standard was devised and published in 2017 by our team with the input from 35 institutes. And the standard, so this minimum information standard for describing patient-derived tumor xenograft models, it consists of several modules and covers about I think it's 63 attributes in total across these modules that are either essential or desirable. So this was a huge community effort. For the minimum information for in vitro models, no standard exists right now. So this was something that we worked on by gathering metadata available across major platforms, providing users with information and models, collecting list of attributes, and then engaging community to collect feedback on these attributes so that we could establish a draft standard, again, with certain modules and essential and desirable attributes. And the scores, model characterization scores for in vitro models are calculated based on this standard that we are planning to publish soon. So back to the project overview. I've talked about getting the data from other platforms, getting the data from the individual contributors. Obviously, there's also, as I mentioned in the beginning, a lot of publications coming out with new models being generated. It is not possible to keep track of everything that comes out. So we try to implement large language models to streamline the process of extracting information about patient-derived cancer models from the scientific publications. So we paired with collaborators at DeepFi, DeepFi project in Boston Children's Hospital, and did a feasibility study of extraction of patient-derived cancer models knowledge from scientific tests using several large language models and using different methods, so direct prompting and soft prompting. If you're interested in the technical details, you can check out preprint that came out last month. But the idea behind this was to see how much information we can extract from the abstracts of the scientific articles, and we can. So now the next step for us is to implement into the pipeline for a more streamlined extraction of the metadata about new models, as well as additional data and knowledge about the models that have already been published, to continue enriching our collection of models and kind of keep the pace of generation, hopefully. So the other thing I would like to mention about our project is that it's all built using the microservices architecture. So each of the components that I have described is a standalone tool, and it can be reused. All the project is open source. You can find this in GitHub, all the code in GitHub, and the Apache 2.0 license. This essentially allows anyone to take the code and deploy a version of cancermodels.org wherever they want, taken that certain minimum requirements for deployment can be fulfilled. This is something that has been done by the EuroPDX consortium, who powered their data portal like this, just specifically for the models within their consortium. And it also can be done by other models as well, again, for deploying the platform itself or reusing any part of the project. A little bit about the future of the project. So we're always we're looking at the development along 3 directions of data, portal development, and software. So in terms of the data, we have we are initiating collaborations with national networks, biobanks, and contract research organizations to increase the number of models and in the resource, especially focusing on those models that are underrepresented. And I mentioned that we have only 10% of pediatric models, 5% of rare cancers, and particularly interested in expanding it to the to include more models from ethnic minority backgrounds. So we're building, we will build on this feasibility study that we've done for the knowledge discovery from publications using large language models, to also ensure that all the published, with the idea that all the published models and studies would feed into our resource. And we're also looking to expand the data types that we currently have in the platform to the to other data types that are associated with these models, such as fusion, methylation, proteomics, as now the models that come these days are characterized with a lot broader with a broader set of techniques, technologies. So we would want to capture that information as well. In terms of the portal development, we're looking into integrating new features and new resources. So we're working on ensuring that model availability is very clearly indicated within the platform. Also adding more model distributors, because for some model distributors, such as ADCC for cell lines, this is kind of, everyone knows about it anyways. But for some models coming from individual academic labs, they are distributed through a smaller companies that might not be very easy to know. So we trying to get in touch with these model providers and to ensure that if they're available somehow through a third-party distributor, we include this information as well to kind of lower the burden on the model provider to answer the email requests of people wanting to get specific models. And we're also working on data visualizations within the platform to provide additional ability to compare models, not just through the list of the search results that we present right now. In terms of the other resource integration, we are working to integrate with the open targets and also clinicaltrials.gov, so to link the existing clinical trials to the models that we have in the platform. And also in terms of the software, we're working on, we're thinking to develop the authenticated instances. Right now, there is, the resource is completely open. You don't have to register. You can view all the data that is available, but there are some use cases where authenticated instances would be of benefit. And also we are thinking about the federation, so the ability to deploy these to deploy the version of the portal within a kind of a, let's say, closed network that would still allow visibility of the models within this network. So if there is an interested in any kind of requirements for model and data sharing, I have my email here, because we are trying our best to build a software that is useful for the users. So we often do user testing and try to gauge the interest and feature requests for this platform through either social media or the presentations or conferences that we go to. And I'm always very happy to hear feedback. If you go on our website, you also see that you can essentially leave feedback on any page of our resource as well. So with that, I would like to finish with the acknowledgement slide. So this project is developed at the EMBL-EBI in the team of Helen Parkinson, and we do it in close collaboration with the Jackson Laboratory, with a team of Carol Bolt. So our team is quite small. It includes myself, our developer Mauricio Martinez, our bioinformatician Tushar Mandloy, and our web developer Marcelo Rios Almanza. We also got some help from members of other teams and also from our collaborators. And our scientific advisory board includes Gail Eckhart, Keith Ligon, Christopher Kemp, Mark Murakami, and Alana Whelm. The funding for this project is from National Cancer Institute of Informatics Technology for Cancer Research. And I also have to thank all the model providers that who worked with us to deposit their data and to go through the refinement of the submission process and everything, essentially through the whole process from kind of our release in February 2023 and through all the refinements that we have been going since then in the last 2 years. We are on LinkedIn and also on Blue Sky, so feel free to join. We'll be very happy. And I'm also very happy to answer any questions that you might have now. Thank you so much.
Thank you for this fantastic presentation. I'll open the, well, you can ask your questions directly to our speaker or write your question on the chat. Well, with that, I mean, actually, I think it's a fantastic platform, really interesting. I have to say, I mean, I'm not a bioinformatician, I'm not an expert, And I think it's really, it seems, and I checked it, your talk, I think it's really comprehensive. It's really easy to use. And actually, I would say it's really necessary because there are plenty of databases out there, but to integrate them all in just one site, it's amazing. So congratulations on this fantastic platform.
Thank you so much. I want to stress that we are integrating multiple platforms, and the platforms that exist, they have a lot of value in themselves because they have the functionality that we might not have, because they're models that come within one large project that have been all processed exactly the same way. They have all the data there, and there are some platforms that allow you to visualize. They have a lot more complicated, comprehensive visualizations of the data, also because the data can be kind of compared across those models. And there's additional analysis that some platforms offer. So and then within those collections, it's very valuable. We are trying to do something slightly different, such as we're trying to kind of gather everything around that is available so that the user who is interested can actually get to that platform, because sometimes unless you know where to look for, you don't know. And that's our aim. That's why I wanted to stress that we always circle back to where the models are available, and we're not taking anyone's credit, just being very, very transparent with what's there. And again, as I said, we're not saying that we can do everything that the other platforms can do, but at least we try to link to additional resources and provide as much information as possible for the user to be able to know what's the next step they have to take, but being able to take this next step with as much information as possible, because that's important to start going in the right direction, so they don't have to turn around when you figure out that something is.
I agree.
Exactly.
Imagine, so for some of our members, imagine that someone has a specific cancer model that would like to offer to integrate it in your platform. So how should he or she do it? How should they approach you to be able to integrate their cancer model in your platform?
Yes, so I will probably, let me just quickly go on the website, because it would be the most informative, as opposed to me speaking it. So this is our platform right now, as it is in the latest release. So we have the submit section, which essentially provides information about data types and formats that we collect. And it also provides this submission templates. So we collect data and metadata through Excel spreadsheets, because based on the user feedback, this was the format that was the most used by the majority of the researchers. So here it's possible to download the submission template to fill out the metadata. There are many of them, both for metadata and data. Again, it's always possible to add onto model, and not every model has all the different types of data listed here. Also in here, there is the metadata dictionary, which essentially explains every single field that we collect across different modules, and the formats, and gives the examples. So that helps in filling out the spreadsheets as well. And also here is the validator, where you can upload this field spreadsheet to get this validation report, and to get the validation report and see whether if it's valid, then the template is ready. We don't have a submission interface where you could actually upload the template that would go directly into the database. So for that, you still need to email us and ask us to submit the model, but it allows quite a bit of, provides quite a bit of information for this early stage of just even understanding what needs to be collected in order to submit the model. And we also have our, of course, frequently asked questions, which is a collection of questions that we got over the 2 years of getting emails through our submission. So we put them out there. We also have some tutorials how to use the metadata validator and the dictionary, as well as essentially kind of the other parts of or kind of the use case of how to search for the models. So this is not so much for the submission. Yes, and also in terms of kind of, this is our open source repositories for anyone who wants to gain the to get the code for this resource.
And maybe I missed it from your talk, but is it possible directly from your web to download the raw data from the different?
So we collect the process data already, so that's not the raw data, not sequence data. So, but we link to the sequence data, which is deposited in the archives, because I mean these days anything that gets published needs to have the raw data deposited to archives. So we actively search for these links, or the providers would give us this link, so we link to the raw data as well. But the raw data would be deposited in the archives that that store the data. For the submitters who, let's say, would like to submit the model that hasn't been published yet, we can help with submitting data to raw data archives that are the EBI, so EGA and ENA, the EBI archives. So we can kind of help with that process as well if someone, you know, asks us for that in the part of the submission process of the models.
For example, to generate representations, graphs, etc., are there different ways of doing it in your web? So in your portal. So sorry, maybe I'm asking really naive questions, but I'm just asking because obviously I'm interested in using it, and it would be nice to know, like, for example, if you can if you have different ways of representing the data, or how can you manage that?
So we're working on some data visualizations right now. Kind of, we provide their more or less of an overview of what's available. So it's in the overview page, just kind of information across all the models. And I mean, obviously, this is also allows you to get the slices of the interested data with specific pre-filtered options. In terms of the data that is within per model, so it's possible to download everything. So in data that, let's say, is here in the molecular data section for each model, the viewing data is usually, it's quite basic right now, essentially through the tables, but it is possible to kind of search through it and also download it for further analysis. So we're working on better visualization of it, but this is also possible to get to the kind of CBio portal for this data, and kind of use the functionality of CBio portal to explore this data within CBio portal. Again, because CBio portal is the tool that is used, I mean, it's been around for more than 20 years, and it's developed really comprehensively, and a tool that a lot of bioinformaticians use. So that's why we decided not to reinvent the wheel, and just to allow this easy access for this model to and visualization with the CBio portal functionalities. We're also working on kind of improving visualization for model treatment. There is currently no really standardized way of displaying this. So this is also something that we work with the Jackson Laboratory on that. But this is a large initiative involving a lot more people and working groups to standardize the presentation of this data. So yeah.
Amen. So, any questions from the audience? Please raise your head or directly raise your hand. Sorry, or directly ask our speaker.
Yes, hi,
Ralph?
This is Ralph Garippa, Sloan Kettering. Really nice presentation. I have two questions for you. One is, we've been hearing about the integration of patient data, both sequence data, for at least 15 years. I think IBM, came out with the software. And this, by and large, has not been successful. What do you attribute the success of the cancermodels.org? Is the timing just right? Is it because you're so inclusive? Is it because the database of patient data along with lab bench-based cell-based data, both 2D and 3D, has gotten so voluminous now? Is that part of the success? That's the first question. The second is, do you ever get a chance to do retrospective, go back and see patients who had a durable remission or patients who unfortunately passed away, perhaps even faster than expected, and see how you can revise your recommendations in the cancermodels.org?
Thank you, Ralph, for this, not easy questions. But great to address. So yes, so why success? So again, I think that the question of integration of data, it's always going to be an ongoing question because there's so many different types of data that, the rate of the data generation and the necessity to integrate multiple data types kind of increases the complexity of the task as we go along. And it's, will be solved to a certain extent, but it's going to be an ongoing problem that we'll have to face. So it's just going to be kind of establish certain, best practices and standards that, the data can be kind of harmonized to enable that as we go along, we can ensure that it will be, interoperable as with the development of the technologies that go on. So this is why kind of it's really important to harmonize and standardize and kind of understand what's important. And there's a lot of initiatives going, working on that. And I mean, so I'm a colleague of the cancer community GA4GH. So GA4GH, Global Alliance for Genomics and Health, works a lot in terms of developing the technical standards and also like policy frameworks to ensure the reuse, ethical, valuable reuse of genomic data and health data. So this is another kind of part that separately to the project that I've described, it is an ongoing initiative that I'm involved in. In terms of the cancer models itself, currently within the scope of the project was to work on the data that comes from the patient-derived cancer models. This is the vision for us and kind of my personal vision to enable to link the data generated from patient-derived models to the patients. And it also kind of a very important task, but challenging task because there are questions that come with, the sensitive data, patient data, and kind of in these, it's very different than, looking at animal or in vitro models. So this is still something that, we would want to expand to with our next kind of rounds of funding and try to ensure these links. The second question was, sorry.
Yeah, do you have a chance to go back after patient outcomes?
Yes, so that's why I started talking about the patients as well. So because right now we only have managed to look at the patient derived models. So we try to focus on the models where we can provide the most comprehensive picture. So we're not necessarily trying to, become the largest resource in the sense that we have the most models, because not every model is as valuable as in terms of how much data is associated with it, and what kind of insights you can get with this data available for the model. So we're trying to see, to focus on the models where we have, let's say, a lot of information available about the patient treatment, what happened to the patient. We try to, we're working on recreating the patient timelines and kind of stepping away from the model to the patient to see kind of what happened with the patient in terms of the treatment and the history, so that this information will be provided alongside with the information from the model, so that then the researchers can use this to extract, insights. And so we're not necessarily going to be the ones looking at kind of what you can extract from this data, but we try to collect and put it together for someone who is interested to look at, retrospectively what could have been done or what lessons can be learned from these particular cases. So this is kind of one of our points in terms of the getting more data into the platform, in terms of trying to have a collection of very, very well characterized models with as much data about the model itself, as well as linked patient data, then can be used for comprehensive studies by our users.
Okay, thank you.
Thank you.
Any other question? Any other question from the audience?
If anyone in the audience is working with patient-derived cancer models and is interested to deposit their models, so please get in touch. Or if you have some other thoughts on how we can improve the resource and what is missing. So we are bound within our kind of scope of the grant that funded our initial project, but we are looking forward to, as I mentioned, make the tool that is useful for the researchers, and we essentially operate based on the user feedback that we receive. So I would be very grateful if you check out our resource and leave comments in the little survey that we have. We have like a 2-question survey that wouldn't take much time, but this is what essentially makes a very big difference for where we move forward next and where we focus our limited efforts and limited team.
Well, thank you, Zina. With this message, I think we can conclude this session. Thank you so much for your time. Thank you so much for this fantastic tool. I'm sure that we are all gonna use it a lot. And and see you all next month. Thank you for joining.
Thank you, John. Thank you, Aaron. Thank you, everyone. Bye.